Today on Osteobites, we're talking with Dr. Richard Gorlick about antibody drug conjugates for osteosarcoma. Welcome to Osteobites. My name is Anne Graham. I'm an osteo warrior and executive director of MIB Agents. It is another gorgeous day today. I love summer. This deserves a great snack. And today I have apples in the hopes that they make me just smarter so I can keep up with Dr. Gorlick and Dr. Wang today on Osteobites. So honored to have Dr. Gorlick back on Osteobites again and as colleague and research fellow, Dr. Yifei Wang. They are talking with us about antibody drug conjugates for osteosarcoma. Dr. Richard Gorlick is Division Head, Department of Pediatrics, Professor, Department of Pediatrics Patient Care, and Director of Department of Pediatrics Sarcoma Research Laboratory, and the lead Division of Sarcoma Medical Oncology, Division of Cancer Medicine, all at the University of Texas MD Anderson Cancer Center in Houston, Texas. His laboratory is the Bone Tumor Resource Laboratory for the Children's Oncology Group, the founding Bone Tumor Resource Laboratory. His molecular pharmacology laboratory is completely focused on osteosarcoma, which is so great. Dr. Gorlick is involved in clinical trials, in part as past chair of the Bone Tumor Disease Committee for COG. He is a past president of the Connective Tissue Oncology Society. Dr. Gorlick has published more than 260 peer-reviewed papers, reviews, and book chapters, and we are grateful that he has assisted MIB Agents as well on our testing and research directory, which is available on our website. Really terrific resource. Thank you for that, Dr. Gorlick. Before we get rolling on ADCs, I need to tell you about the many individuals and vaccinated teams who are getting together in real life to outrun, outwalk, or outpedal osteosarcoma. You can do this virtually as well. Dr. Trucco and the Cleveland Clinic Solid Squad are the biggest medical team on our roster. So, any other medical team can join as well. Not to, get you all competitive or anything, but, just saying. You and your team can participate anytime from now through August 31st with a 5K, a half marathon, or a full marathon, or a combination. Treadmills and Pelotons count too. We are grateful for the sponsorship of BTG Specialty Pharmaceuticals for outrunning osteosarcoma. You can join or create a team at mibagents.org/outrunning. You will also get some serious swag for registering, including the must-have t-shirt style of the season, an Outrunning Osteosarcoma t-shirt. The links will be shared on this podcast and on YouTube, where all Osteobites and MIB Agents videos live. Dr. Gorlick, would you get the introduction started, please?
Yeah, no, thank you very much for that kind introduction. Indeed, I've spent my whole career working on osteosarcoma. My snack isn't nearly as good, like potato chips, just doesn't rank with apples, but it's really a pleasure to come back to Osteobites. I am glad you're having one of these kind of events, like one of these things where you're encouraging competition across groups. Physicians, indeed, even though we work together, we are a bit of a competitive bunch, but I want to sort of say that there's a disadvantage for the outdoor activities in Houston in July and August. So, I was glad you had a Peloton option and a treadmill option, because for us, being outside this time of year is a little bit hot. For really this presentation is coming about largely based on a presentation made at the American Association of Cancer Research. And the reality of it is, us as principal investigators who lead the lab get a lot of the credit, but actually at the AACR meeting, it was Dr. Wang who gave the talk, largely because Yifei is the person who did all of the work. And, scientists know when you really want to actually figure out what the details are and what exactly was done, it's actually the people like Yifei who know it better than anyone else. Yifei, can you introduce yourself?
Yeah, sure. I'm Yifei Wang. I'm a postdoc in Dr. Gorlick's lab. And before I joined the lab, I was an orthopedic oncologist back in China.
Well, hi, everybody. My name is Mia Sandino. I am a junior board member for MIB. I have been an osteo warrior for almost 3 years now. And my snack today is a fruit crumble, which is actually surprisingly good. Not really a crumble person, so I'm actually very much enjoying it. And I am currently transferring from the University of Washington to the University of California, Santa Barbara, total mouthful, as a psychological and brain sciences major.
Hi, I'm Shannon McCormack. I'm also a junior board member. My connection to osteosarcoma is my older brother had it, and I don't have a snack. I had a bagel before this, so really slacking off. Sorry, guys, but I'm happy to be here and excited to see what they have to share.
Okay, let me start sharing some slides, and I'm going to start as they pull up the slides. Start by making an initial statement that, you know, I always use my slides a little bit as a means of like figuring out what I'm going to sort of talk about in general, keeping the slides complicated so that folks can get all the details that they may want, but at the same time, not talking through all the details because it starts bogging people down. With that, we're going to be talking about new drugs, a new category of kind of drugs called antibody drug conjugates that Yifei has done a lot of work on finding ones that apply and can be developed for osteosarcoma. You know, every good start, you always have to give a little bit of an introduction and just sort of talk a little bit about the disease. This is very, very general information, but basically gets at, you know, osteosarcoma is really rare. The graph that's in the middle of the slide shows you age by decades of life, and then the size of the bar. Going up is the number of girls, and going down is the number of boys that get osteosarcoma each year. And what this is emphasizing is that is what you already know. It's young adults and teenagers who are by far the group that's most affected by osteosarcoma. It remains a rare disease, but at the same time, that is the group that, you know, gets it. You know, you guys sometimes see your x-rays, but the osteosarcoma on an x-ray, because it makes bone, has a very characteristic appearance, you know, referred to as a sunburst, and that's the picture on the top right. And the biggest challenge is when it spreads to other parts of the body, particularly the lungs, which you'll see as dots on a CAT scan, which is, you know, what you're seeing in the bottom panel on the right. So that's just the sort of general starting point for osteosarcoma, which I know you know about. Then from there, you know, my laboratory for its entire existence is focused on osteosarcoma. And a lot of what we've tried to do is try to figure out drugs that had been developed predominantly for adults that may apply to osteosarcoma. Some of these are drugs that had been developed for a variety of different purposes, almost never for osteosarcoma, but for different cancers. And these are things that you know, was sort of our approach in the past. And, you know, it was really what drove our interest in osteo was the availability of the drug because it worked in an adult cancer. And, you know, unfortunately, while some of these trials had a bit of activity, none of them had the activity that would have really sort of said, this is a win for sure. And again, some of these are antibody drug conjugates, and I'm going to talk about that a little bit more. And really, I think the challenge that you may have heard is, you know, osteosarcoma is a disease where we cure a lot of the patients, but unfortunately, if you look back over the last couple of decades, it's not really getting better. And, you know, part of the reason we want you guys all to advocate for research and, you know, support research is because research is the path to making things better. And that's really, you know, our emphasis of discussion today. And really what this is trying to sort of illustrate is we have evolved how we think about developing drugs for osteosarcoma. We went from an approach of starting with drugs that are available to instead trying to identify what would work best in osteosarcoma. If that drug exists out there, that's great. But instead of relying on what's already made, if that drug isn't out there, maybe we actually have to start making drugs specifically for osteosarcoma. And again, you know, this sort of continues the introduction where we know there's some surface proteins already. We know those surface proteins can be targeted in different ways. We know, you know, cell therapy approaches and antibody drug conjugates are ways of approaching targeting osteosarcoma, but at the same time, we don't think there has been a lot of attention on finding what's the best target in osteosarcoma, and that really has driven the work of today. Part of the reason you can start talking about making a drug for osteosarcoma is the reality of it is making a drug targeted by an antibody is easier than making a drug from scratch. Making an antibody, there's ways to get immune systems to sort of just through proven laboratory methodologies to make antibodies to a foreign protein. That's how you get immunized to something. If we can make antibodies to something, we can then bind it to a drug and try to use that as a treatment. And what this slide is showing you is that, you know, antibody drug conjugates were kind of rare before the 1990s, but they're exploding in number, again, for the reason I described. They're relatively easy to make. There's known ways to make antibodies. There's known drugs that can be combined with them. And we just look, more and more drugs are getting approved for different indications. I will highlight, none of these are osteosarcoma, but it's an approach to make drugs quickly. So the whole defining hypothesis for this was surface proteins. And we thought because osteosarcoma makes bone, some of these may be related to like a bone differentiation, that we could target these therapeutically. I am a lifetime New Yorker who only moved to Texas at the end of 2016. Part of the reason I moved to Texas, to be honest, is MD Anderson. And part of the reason MD Anderson does as well as it does is because the state of Texas has a program called CPRET. This is a long acronym for, you know, the Cancer Prevention Research Institute of Texas. It is basically a state-funded initiative. It is the largest state-funded cancer research funding enterprise. So we were very fortunate to, early after I arrived at MD Anderson, to get a grant from CPRET that allows us to profile all pediatric solid tumors, but a lot of what we see at MD Anderson is patients with sarcomas. So a lot of these are osteosarcoma patients. And this is where I start, you know, heading down the path of, this is all Yifei, none of this is me. This was sort of the pipeline he decided to create. So what he did is he started to take a series of models of osteosarcoma and actually use a comprehensive technique, you know, again, you know, quantitative mass spectrometry, to basically look at every protein that's expressed on the surface of those models, and then actually look at what are the most common. He started integrating that with databases that exist in the public domain, used you know, software to help narrow it down to surface proteins, came up with a list of therapeutic targets and started validating them. And we're going to talk more about that work. And actually what finally led to this abstract was the first drug that nobody really knew would work for osteosarcoma, was tested in osteosarcoma models and had encouraging results. Now I could skip the whole talk because you've heard the whole talk. Not really, we'll show the data. You know, obviously some of these things, this starts with the mass spectrometry and, starts sort of showing you how it was narrowed down to a series of genes that were viewed as the most promising targets. We're going to talk more about those 4 particular targets. And then we're going to talk about some more targets that, we're in the midst of investigating or working on developing drugs too. So one of the things that was done very soon after identifying these 4 promising targets, and again, I'm going to talk about these a little bit longer, was we made a comparison of the expression in osteosarcoma, which is the red box, versus the expression in blue in a series of normal tissues. If we wanted the drug to sort of work best, we wanted to have the most expression in the red box and the least expression in the blue boxes. And some of them, even though they look somewhat close, are already drugs. So that sort of tells us the side effects aren't going to be so bad for these. But I also want you to notice the difference between in the red and the blue, because we're going to come back to these types of graphs later. So this is exactly, where it goes next. So MRC2, MMP14, CD276, and LRC15 have already been used for targets for antibody drug conjugates. We know these are things that can be given to people and have acceptable side effects. A couple of them, specifically the LRCC15, is actually, even though we found it as a target, that had been a subject of a trial in Boston, actually for osteosarcoma. So some of these had made it to osteosarcoma, but most of them hadn't. And like I said, the MMP14, nobody really thought about as an osteosarcoma drug. And so one of the things you do whenever you start doing things as data one way is you try to verify it another way. I know it's painful to the postdoctoral fellows, but the key to life is verification. Any one result, you just don't know why it happens. That's why we believe in clinical trials and testing big groups of patients. And unfortunately, we're thorough. So Yifei had to do this multiple different ways for each of the models. And you're seeing flow cytometry data and Western blots. And, again, I don't want to make people crazy, but, it's actually easy to understand blots. And if it's dark, a blob, it's there. So you see all of the dark blobs, it basically tells you it's there. And the bottom one, when the color is to the right of the gray, that means it's also there. So all this is showing you is the data that shows the proteins there. That said, the caveat for this is again, on models and cell lines, this isn't on patient samples. So again, being thorough, one of the things we have is always tumor tissue left over from patients who are willing to allow us to use it for research. We get slides from their tumor. It doesn't consume all that's there. This is always done with permission so that folks know we're using this for research. And this is an example of the type of study we did. And what it's showing you in this particular type of thing is called immunohistochemistry. And here, when things are brown, it means the protein is present. When things are blue, it means they're not. When it's blue, you're still seeing the nucleus. It doesn't mean the experiment's wrong. It's just not the protein you're looking for. And what you're seeing in the leftmost column is actually patient samples compared to the model systems, compared to something that's negative. And what you're seeing is the patient samples are maybe a little less brown than the PDX tissue, but they're pretty brown, certainly nothing like the negative control. Each one of those boxes, though, is only one patient. So what we do is we do these on a lot of patients, and those are the tables you're seeing on the right. So when you see those numbers, 6, 7, and 24, that's the number of patients for MMP14 who had, middle they're almost the highest and the highest level of protein expression. None had none or very few cells positive. And in the bottom part, it tells you how brown it is. So this really validated this is in samples and this is a real finding. So this started driving, okay, why don't we try to look at, some of these targets, get the actual antibody drug conjugates, and actually start testing them in, again, model systems that can look at whether the drugs actually work. This has already been published in Clinical Cancer Research, but and CD276, which is also known by the same name, by a different name, P7H3, worked in all of the models using different ways of targeting this. We can talk about this a little bit more, but Seattle already has a clinical trial of CAR T cells for B7H3 underway. And that, I believe, includes osteosarcoma patients as being potentially eligible. And again, there's other drugs that are, at this point, tested and reported and have similar activity. So this is really what got presented at AACR. There is a drug called BT1769 made by a pharmaceutical company by the name of Bicycle Therapeutics. This is a company that was developing it for a variety of cancers, but they really did not have an osteosarcoma development program. The work supported that this is a target in osteosarcoma, and the testing in the model systems shows this drug actually really worked. Where COG and others are discussing with Bicycle Therapeutics potential trial. Companies have to fit it into their portfolio of developing drugs for other things. So I can't give you a precise time a trial will happen, but I'm confident a trial will occur. People who are lab scientists, they always want to show, part of the reason there's an advantage to talking rather than just reading is it takes a long time to go from, submitting an abstract, which is months before the meeting, presenting the abstract, and then people starting to hear the result, let alone starting to see it in publication. And the Bicycle Therapeutics work was the work presented at AACR. So we always want to show the work that hasn't been shown yet. So this is a drug that's made by a company in, I'm going to blow this. I am so bad. It's Denmark, Sweden, or Norway?
Denmark.
Denmark. I know it was one of the Scandinavian countries. Thank you, Yifei. I told you he does all the work. So this is, again, another target that came out of the same screen. This company is an earlier stage company. This drug is not in trials for anything yet. They are thinking about doing a trial for osteosarcoma. To be honest, they have never really thought about doing an osteosarcoma trial. We asked them for their drug because it sort of came out of our screen. And just like the Bicycle Therapeutics drug, it seems to work pretty well also. So we're excited about this. Again, there's a lot of curves on this that don't look as good as the others. To be honest, they were earlier in drug development. So some of the drugs were made with the same antibody, but different drugs attached. And that allows us to have some input into what drug they add to the antibody, which may have some advantages. And we have other proteins, and these proteins are at various stages of sort of being characterized. But what as I promised, I wanted to come back to that blue and red graph. So these two proteins are proteins that don't even have drugs. They're drugs where the like nobody has made an ADC to them. What we're highlighting in the middle panel is the separation here between the blue and the red, and this is relying on your memory a little bit, is even greater than the drugs that are out there. So that offers the promise that maybe the drugs will have even less side effects because there's less normal tissue expression relative to the expression in osteosarcoma. And we're actually pursuing making antibodies to these proteins as a first step to making the drug for osteosarcoma. Other ones, we were kind of lucky. We had one protein where, again, there's no drug out there, but this particular protein, there was an antibody out there. And so we were able to sort of look at protein expression. This one is giving us a little bit of a jump start because what we're able to do is acquire the antibody and immediately use that to make a drug for osteosarcoma. And we are working on that actively. And again, this shows you just so, you know, again, there's a lot of steps in making sure things work as a drug. You know, when the antibody binds, the graph on the left is showing you that, not the graph, the figure on the left is showing you it has to be internalized. The graph on the right basically shows you this antibody is internalized. I'm not going to fight to make this video go, but you know, there's a video, or is that just an image? Yifei, help me out.
Yeah, it's a video.
Okay, let's make the video go. I like videos. What does that show?
Yeah, so when the cells start to have the orange color, means the antibody is internalized by the cancer cell.
Okay, so you're watching the cancer cells eat the antibody. That would be the way I would explain that. That's kind of cool. I must admit, I've never hit play on the video. All right. And, you know, again, I never, you know, I think we have a history of things in osteosarcoma. You know, there's been a lot of great ideas, and I'm going to sort of harken back. And I'm sorry that it has to be a little bit of pessimism. Like, we've tried a lot of things that don't work. And so we have to think about, even as we do this, why is this going to fail? We think it's going to be a really big advance, but if it's going to fail, why is it going to fail? And one of the things that we started doing was to figure out how our models can become resistant to some of these treatments, meaning how can, you know, despite them working initially, would sometimes they stop working. And you know, what you're seeing in the panel left is, you know, we could actually, if we observe long enough and treat in particular ways, we can see this particular drug work and then not work. And one of the reasons that is, and this is what it's showing you, is that, you know, with treatment, you know, the expression of the target can be lost. And usually what that means is actually the cells who had it were killed, but there were some cells that didn't, and those were able to replace the ones that did. You know, again, I say this, you know, and I'm sorry, there's a pessimistic note, but knowing how things don't work actually allows you to anticipate that. Even in therapies that have been dramatically effective, this sometimes happens. And the answer to that is usually to treat in combinations. Treat with more than one antibody at once. Treat with multiple at once. And so even though we have to start by sort of giving patients these one at a time, I think the goal is going to be very quickly to giving multiple at a time, so that we don't see these type of phenomena. So the goal is curing everybody always. So really, in a very quick period of time, I think I've covered what was presented. We know that CD276 and LRCC15 are already been in clinical trials. LRCC15 and osteosarcoma specifically. There were some side effects, so that's that may need some revision before it comes back. CD276, other folks are developing trials of it. I did mention that there's a CAR T trial of that available in Seattle, but there's trials in development here as well as at St. Jude as well. MMP14 is the new one. We're working on that. Hopefully that'll be in trials soon. It is in trials for other indications. And so when I say trials, trials in osteosarcoma. MRC2, there's a drug. I think, if the results continue to be promising, we're going to hope to be able to get that to patients soon because that's already been made. Surface protein C, we're going to make the drug. And proteins P and I, we're going to work on making the antibodies so we can make the drug. To us, this almost looks like a pipeline that can have multiple different drugs available. We think giving them together is going to ultimately be key. How we work this into like standard therapy, we're going to have to sort of take some time to think about, but we think these are exciting developments. And so we have a lot of opportunities for impact. We think we've made a lot of progress, but we think a lot more needs to be done. I think this is mainly meant to say we have a lot more work to do. We are committed to doing it, and obviously appreciate partnering with you guys. Obviously providing samples, all these type of things have been invaluable to our work throughout. And I can never stop without thanking actually the people who fund our work. We've been very fortunate to have grants from the NCI and CPRIT, but a number of foundations have supported us as well. Clearly, I gave the talk, but I do none of the work. My lab is a big team. Yifei obviously was the key person in all of this work, but the other folks helped them on parts of it. We do operate as a team, and thank you guys for the opportunity to speak to you guys today. So thank you.
Thank you. And thank you, Yifei. Even though I know, Dr. Gorlick, you're being modest. I know you do a lot of work. And one thing that always astonishes me as coming from the patient side is every time we do an osteobites and there's funded research, it is so frequently funded by nonprofit organizations and, outside of government funding, like really, really important to participate, and not just MIB's funding mechanisms for funding research, but it's really important that if it's not MIB, you find an organization that meaningfully funds osteosarcoma specific research and that the money goes to where it's supposed to go. And if it is an osteosarcoma specific organization like Osteosarcoma Institute, MIB, we have a big why we want to fund osteosarcoma specific research. So thanks to all those organizations that are helping. And oh my gosh, do we need more? We need more.
And I would just extend the comment that that's a real problem for rare tumors. So the biggest funder of cancer research in the United States is the government. So the National Institutes of Health, and specifically the National Cancer Institute. And the way they score grants, and to be honest, advocates sometimes get involved in the scoring process, and even in the review panels, and I would encourage you guys to do so if that's something you'd be willing to do. And really what it gets at is the fundamental score. What's supposed to drive it is a word called impact. How much impact is this going to make? How much is it going to change the world? And while that's a great statement, and I believe in it, what happens in rare tumors is they turn around and say, well, guys, this just isn't a lot of people. So your impact just isn't there. So what it means is the hurdle, and we've been successful at getting some NCI funding. The hurdle is always, there's just, in their view, not a lot of people affected. So your science has to be beyond, if you're talking about the quality of science working in a common cancer like colon cancer or a rare cancer like osteo, our science has to be better to match the impact because they have numbers. I don't know. I care a lot about osteosarcoma. I see patients with osteosarcoma. The people I see care about it. You always have to sort of make clear that, this is impact, guys. You are impact.
Yeah, again, like on the patient side, and having attended 3 funerals in the last 6 weeks for kids 18 and under, we just have to make it better. Okay, we have some questions.
So this is in the context of protein C. Is protein C the same protein that when it's low can cause blood clots?
Cells stick together to form tissues. It's one of the proteins involved in cells sticking together, and not involved in clotting at all.
How can non-osteo warriors like parents, other family members, friends, assist in your work and further research?
That's a great question. So for us, I think one thing that we're always looking to do is sort of create awareness around clinical trials. I think, one of the things that I've always talked about is, just keeping a list in folks' minds of what's available in different places. I think there are a lot of clinical trials out there, and I view them as all good because, okay, so I want to take a step back. In a different context, I gave actually your guys' group meeting, I gave a talk, an introduction to others speaking about clinical trials. And I am an immense believer in clinical trials. I think if we treat people in organized ways, we learn from the experience of each person to make it better potentially for the next. To me, clinical trials aren't using people as a guinea pig. It's just being systematic about how we think about things. In a clinical trial, generally we're looking to give the treatment I would want to give you outside of a clinical trial, but doing in a structured way that allows us to learn from it. And to me, that's the definition of it. So I think clinical trials everywhere are good. This is not about the clinical trials at our place versus another's. I think every place's clinical trials are good. All of us have finite resources, and we can't investigate everything at every place. So what that kind of means is that, a group of us are going to do one, a group of us are going to do another. They're all overlapping. They're all over the place. I think the key is for folks to get what works for them, what's close, what's available, what sort of matches what they've had before, so that it's what their doctor thinks has the most, without data, the most chance of working. And I think doing that in an organized way is sort of critical to our future. On the prior osteobites, we haven't carried forward. I think for patients with osteosarcoma where it's come back, I think having an organized way to get, multiple opinions from us in a simple way would be a great thing. We talked about that being a physical thing, but I will highlight Dr. Trucco put together a tumor board to pull us together to at least have the conversations. So there's a lot of ways to do this. And to me, be educated, be aware, be connected to your community, encourage learning, encourage awareness, and all of those things are, to me, what's most important.
What stage of treatment do you need to be in for this process if chemo doesn't work?
Everybody with high-grade osteosarcoma, unfortunately, needs 2 forms of treatment. Everybody needs something that is directed at the site where the tumor started, which for osteosarcoma is most of the time surgery. And the problem is, we know no matter how early we catch high-grade osteosarcoma, and I'm throwing in the word high-grade because there's occasional low-grade types which aren't the same, and that's not most of osteo in kids or adults for that matter. So every high-grade osteo, unfortunately, even if we see their lungs are clear, we know without systemic treatment, we're going to see lung nodules eventually. That said, there's a very standard cocktail for treating osteosarcoma, and it's a lot of the drugs you guys have gotten, methotrexate, doxorubicin, platinum. You know, every once in a while, there'll be a trial to try to optimize those, but those take a lot of us and usually get done through cooperative groups because you're only going to see small changes because the treatment works to begin with. The trials that I'm talking about are, unfortunately, when those drugs fail patients and their osteosarcoma comes back, the treatment choices aren't as good. And that's where there's more variability. And a lot of us are trying a lot of different things to try to make that better. There are some standard treatment options, but that's where you got to think about, you know, what's standard, which can be more chemotherapy. It's certainly more surgery if it's operable. But what else may change the odds? And that's where trials become more important.
Before we go to the next question, I just want to mention that we have on our website a really cool little platform within our website where you can find an osteosarcoma specific trial in less than 60 seconds. And it's connected to clinicaltrials.gov. It's the same trial information, but it's specific to osteosarcoma, and you can find a clinical trial in under 60 seconds. It's a service that we pay for, but it's a way that we can get clinical trials, patients to clinical trials, which, as Dr. Gorlick says, is incredibly important and so needed. Dr. John Healy is on board and has a question for you.
Hey, Richard, thank you very much. Outstanding as always. And I would just reiterate some of these comments that even technologically retarded people such as I could find the trials on that site. So thank you. Just another comment about trials that has been shown in pretty much every disease, even the people where the trial agent may fail them. And I like your terminology and expression on that, by the way, rather than that the patients are failing the trial is failing them, is the proper way to put it. But even those other people, because they're being studied systematically and involved in a systematic program, do better than they would have done otherwise. And so that's been seen time and time again. So I share the thoughts and encouraging people to participate for their own benefit as well as for others. But you know, as you know, I've long had an interest in the other aspects of the local tumor microenvironment, including things like pressure in tumors and the physical chemical barriers to particularly large molecules like antibodies, as opposed to small molecules. And so showing how well things work in culture is different than, and you did show some PDX model activity, for example. But even that's very difficult because you have xenograft situations, and how to investigate immune therapies in xenografts is problematic. But exclusive of that, you know, do the drugs penetrate solid tumors, and are there ways to enhance that? And you referred to combinations of antibody therapy, immune therapies, which are the rage in most carcinomas presently. But do you think combinations of small molecules and antibodies, you know, what is their prospect?
So I have to start by saying, one, thank you, Dr. Healy, for being on this call. For those of you who don't know, Dr. Healy was an immense mentor from the first decade of my career, along with Paul Myers at Memorial Sloan-Kettering. So these are wonderful friends and teachers. Of course, Dr. Healy and I are always on the same page because I learned from him. So I'm going to just sort of say we're aligned around that. I think you're absolutely right. What we're getting into is osteosarcoma is both heterogeneous, meaning even within one person's tumor, there's a bit of variability. And then from there, there's like a lot of like whole blood flow dynamics that can influence things. You know, particularly in the context of things like cell therapy. One of the biggest challenges for cell therapy is actually the cells getting to where they need to get to. Like if the cells don't get to where they have to kill the tumor because they're big, they're not going to do what you want them to do. And again, I think you have to do things stepwise, but there are ways to try to attract tumors, as an example, to where they are. You know, some of them are, if we think about cell therapy, there's all these chemicals called interleukins, which are part of our inflammatory response that drive the signaling. There's a strategy that sort of exists at MD Anderson where you use actually one cell therapy targeting the tumor as a way of attracting other cells that also target the tumor to the same vicinity. It uses a protein cell surface vimentin, which usually isn't on the cell surface, to drive the chemoattractant to the sites of the tumor. What also makes this complicated is some of the whole dynamic issues are different if we're talking about a leg and bone versus leg in your lungs. The blood flow dynamics there aren't the same. We have to make sure that, you know, whatever we treat with treats the whole body. We know, you know, osteosarcoma can come back in other bones, and things can sort of come back that way too. We know there's other ways of manipulating things to have them work together. Immune checkpoint inhibitors. They have not, these are drugs that stop the immune system's break on recognizing cancer cells. As single agents, they've had some activity in sarcomas, but we would like it to be more. Combining cell therapy approaches with immune checkpoint approaches is going to be another way of augmenting activity. It's kind of what I was saying with those resistance panels. You know, Dr. Janeway and I have conversations about osteosarcoma, another colleague and friend. And we always talk about our shared belief that a single drug's never going to be the answer for osteosarcoma. Everything has to be about combinations because of the complexity and a lot of these issues. So I think Dr. Healy is absolutely right. Like I said, he taught me. These things have to evolve to combinations that address whole organism issues, meaning blood flow, pressures, as well as address issues of, you know, just things getting where they need to be. So there's a lot to be worked out. I think we've made a lot of progress. We're really hopeful about what's been done, but please, there's a lot more work to be done. Now I'm worried like next Paul Myers will come on and then Joe Bertino, and that will be my whole mentorship life.
Well, I'm glad I was first then.
Yeah, no.
Because in that I was actually last. So they had already.
That's actually, but in very short order. So it was Paul Myers first, then I went to Joe Bertino's lab, yes, and then I became interested in osteosarcoma. We were talking about a 2-year span. So third, really, but in 2 years. Yes. And then I spent another 10 years there.
I have a question about the first line of treatment for osteosarcoma and why symptomatic therapy can lead to relapse in approximately 30% of cases. Why is it not targeted therapy used in the first line of treatment and not in relapse?
We're a little bit like the legal system. The legal system evolves largely based on precedent. Like if you listen to, you know, I'm not a lawyer, but you know, I read the New York Times, and when I hear about a Supreme Court decision, it's always, how do the prior decisions of the court inform how I think about the present case? And things evolve, but you know, it's a lot about, you know, how things have worked in the past. You know, over the last 30 years, I kind of highlighted there hasn't been a lot of progress, but please don't take that as meaning there haven't been trials conducted throughout. We've tried so many different things, giving more chemotherapy prior to surgery, giving more therapy, adding drugs. There hasn't been, if we go back 30 years, there weren't targeted drugs, but we added other kinds of drugs, immune approaches. And the problem is it didn't change outcome. So even though it sounds like it should have, and those trials were all good ideas at the time they happened, at the end of the day, no matter what you do, that outcomes kind of stays the same. If I had a choice of giving more to get the same or giving less to get the same, I'd rather give as little as possible. And that even leads to the question, have they ever tried to give less? And the answer is they did that too. And when you give less, unfortunately, the outcomes become worse. There was a big study from a group in Germany, the German cooperative group, CAS. So it wasn't a group in Germany, it was all of Germany. And outcomes got worse. So I don't know why those, drugs are the right ones, but, the trying other things has led to them to continue to be the best. And part of the reason I keep emphasizing 3 drugs is there's actually some variation. So the 3 drugs that we typically use in America are methotrexate, cisplatin, and doxorubicin. The reality of it is some places swap out the methotrexate for ifosfamide, some places swap out the cisplatin for the ifosfamide. So there's actually 5 drugs, but you have to get 3 of them. And part of the reason the North American standard is what it is because of the side effects of those 3 drugs versus the others. As terrible as those drugs are, some of them have less long-term consequences. And that's what North American doctors have prioritized.
Dr. Wang, what are your thoughts on that kind of international collaboration? Like if we're always talking about tumor donation and biosamples and using the Osteosarcoma project, and it seems like more is more, more is better on this level. And is there an opportunity for international collaboration on this level?
Just like you said, more is always better. So I think like surgeons and the doctors in China would love to contribute more to the Osteosarcoma Society, maybe to help to enlarge the tissue bank and participate in multiple clinical trials, just to try to move everything forward. That's what I understand from my experience.
Yeah, how do we do that?
Wow, I am. I was just a junior surgeon, and I'm just a trainee here in MD Anderson, so I'm not sure I'm the right person to answer that big question. Sorry.
Yeah, our motto is Noah built the ark, and the experts built the Titanic. So we believe in the amateurs making things happen. And we've started a collaboration with Shiyu Children's Foundation. We have a book, Osteosarcoma From Our Families to Yours, and it's available in China now. If you get osteosarcoma in China, you get the book. And it seems like we could take the next step and share more tumor samples, even if it's, it's the data, just the data. Oh my gosh, just the data. Okay.
Yeah, no, I think that's a great idea. Obviously, we'd be supportive.
Certainly on an individual basis, there is no pride that influences this, and it's an effort to help people. And we're delighted to share whatever insight or knowledge we have, and it's a privilege to receive a call. In this COVID era, with suspension, emergency orders suspending the regulations that limit communication between doctors and patients in different states, that's enabled us to do telemedicine consultation with people. Now, the problem is, and it's great that COVID's are coming under control. The bad parts that they're reinstating these limitations that are inherent in our republic of individual state licensure and that sort of thing. So it impedes this. But I can do these consults and as long as I don't charge. So I do them for nothing so that it gets around the legal system. But it puts me at a little bit of vulnerability in the situation. And hopefully momentarily my license in Texas is going to come through. I'll be available to discuss things with patients and actually my own house down there.
Very nice.
But I'll have a license shortly, and so that's great. So it is conducive to improving communication throughout the United States. We're greatly limited, however. The European Union has these very onerous privacy laws and regulations, and so it doomed this randomized surgical trial that I just completed. But I had people lined up in Austria, Netherlands, Germany to participate in this, which is hard to do, to randomize surgery. Surgeons are just not raised that way in their mental and emotional status. But that was lined up, but it was blocked by these regulations regarding international sharing of data in this regard. And so no one in my institution was not willing to pay the bill because of the legal risk of sharing the data for this randomized surgical trial that was funded. I mean, this is otherwise funded, but I didn't include millions of dollars of legal risk liability insurance for this. So there are, as much as there's goodwill on the part of the patients, the families, the foundations, the doctors, there are still these impediments, unfortunately. And so these are things that I think are important to raise with legislators and regulators and things to try to beat down those obstacles to us trying to do good work and raise everybody's boat.
Agreed. It's a great conversation and we need to do more. We need more conversations. In the meantime, I'll wrap it up by saying thank you so much to everyone who participated in our outbidding osteosarcoma auction. We had some big celebrity musicians, athletes, artists, luxury hotels, and style makers. We had so much fun with it. I cannot believe the cool participation we got. And oh my gosh, it was really so fun and really amazing to get people outside of osteosarcoma in the celebrity and art world involved. It was really cool, so great. It was really a great success, thanks to the generosity of, well, everyone. So, so big thanks for that. And that helps us to do the work we do and fund research. The next 2 weeks, we're taking a break for 4th of July weekend, and then I have a short vacation, and then we'll be back after that. In the meantime, thanks to our guest, Dr. Richard Gorlick, who works with astonishing tenacity to obliterate osteosarcoma. Thank you, Dr. Wang, and our junior board panelists, Shannon and Mia, our sparkling panelists, whom I adore. And thank you for joining us today, and we'll see you in a few weeks. Thanks, everybody.
Thank you so much.